Greetings, my name is Ander Roca, postdoc at the Lione Pathology Institute Everest, and today we will present your recently published article in which we characterize the TLR8 agonist selgantolimod as a potential strategy against chronic hepatitis B. In this context, selgantolimod has been reported as safe and well tolerated during its clinical evaluation. Moreover, our collaborators had previously described that selgantolimod induces the production of a wide variety of inflammatory cytokines in human PBMCs, which activate multiple immune mediators. However, little was known regarding its effect on immune responses within the liver microenvironment. Thus, this was our aim. As a first step, we analyzed publicly available single-cell RNA-seq data from human liver tissues in order to identify populations that express TLR8 and are therefore susceptible to the direct action of selgantolimod. This allows us to observe that TLR8 is expressed mainly in the myeloid compartment, from which we focus on the characterization of liver-resident macrophages or Kupffer cells. With this goal, we also took into account that CD163 has been described as a useful Kupffer cell marker and thus optimized a protocol for the isolation of CD163 positive cells from human liver resections. A detailed description of this method can be found in a recent STAR protocols publication. Having these cells, we treated them with selgantolimod and studied its effect by performing RNA-seq. This allows us to observe a particular transcriptomic profile characterized by the downregulation of many genes that give Kupffer cells their identity, including transcription factors such as ID3 and SPICY. Moreover, we were able to validate that these transcriptomic changes also take place in vivo, as observed in the liver of macaques treated with increasing concentrations of TLR8 agonist, showing that selgantolimod is able to modulate Kupffer self-differentiation status. In parallel to this, and as expected, selgantolimod induced a series of gene expression changes associated with inflammatory responses in human Kupffer cells, as well as in liver tissues from macaques treated with TLR8 agonist. Changes that translated into the production of cytokines such as IL-2, TNF-alpha, and IL-6. Next, by treating human hepatocytes with this Kupffer cell conditioned media produced in response to selgantolimod and performing RNA-seq, we were able to observe a marked decrease in the expression of NTCP, which, as you know, is one of the main host factors implicated in HBV entry. And indeed, when we treated hepatocytes with these cytokines produced by Kupffer cells and then infected them with HBV, we were able to observe a significant reduction of viral parameters such as HBs antigen, HBV RNAs, and cccDNA, further supporting the notion that selgantolimod has an impact over HBV entry. And finally, the use of an antibody neutralizing the effect of IL-6 in the supernatant was able to reverse this decrease of viral parameters, showing that selgantolimod indirectly impairs HBV entry via an IL-6 dependent mechanism. So just to conclude, we have shown how selgantolimod triggers Kupffer cell plasticity to promote the expression of antiviral programs over those associated with the Kupffer cell identity, leading to an impaired HBV entry via the IL-6 dependent downregulation of NTCP. These observations have relevant implications for the identification of intrahepatic biomarkers associated with innate immune activation. Moreover, taking into account first its previously described modulatory action of adaptive responses in context of an established HBV infection, and second, our results showing an effect over de novo infection of hepatocytes, this suggests that selgantolimod and its dual mechanism may be an interesting asset for the clinical development of combination therapies against chronic hepatitis B. So, I would just like to thank all of the members from the Lione Pathology Institute Everest, our collaborators, funding agencies, God for this opportunity, and of course you for your interest in our work. Thank you very much again and see you next time.